Good. So now comes the second part, so to say, of the story. Why do we need other information than only macroscopy? Also, it helps us a lot. And other information here, I would like to refer to the cytoarchitectonic segregation of the cerebral cortex in particular. And cytoarchitecture means the distribution of cell bodies, the arrangement, the presence of particular cells. And the cytoarchitecture has been subject of analysis of Corbinian Brodmann, who published in 1909 a very famous map. And this is a map, as you have seen it very frequently, it's only color-coded to better show you that he has distinguished more than 40 different cortical areas in the cerebral cortex based on the distribution of cell bodies. And the image below, coming from the triangular part of the inferior frontal gyrus, shows the distribution of cell bodies in such a way that they are arranged in different layers, but also in different columns. And there are particular cells somewhere present which are particularly large, here for instance, or there. And if you have a look to another cortical area, then, for instance, here in the occipital region, the primary visual cortex area 17 and the secondary visual cortex area 18, the pattern of the distribution of cells looks simply quite different. And these different patterns of distribution of cells, that is different cytoarchitectures, they are indicative on functions. And although at that time it could not be demonstrated, Brodmann was convinced that every cytoarchitectonic area subserves a different type of brain functions. So when we have come back to what Svenja Kaspers has shown just recently, there are, of course, functional areas that are associated very closely to sulci, like, for instance, the primary motor cortex, which is located at the central sulcus, more precisely anterior to it, or the primary auditory cortex, which is located at the Heschel gyrus, or the primary visual cortex, which is located somewhere where the calcarine sulcus is. But there's a large amount of so-called higher associative areas, multimodal areas, which are not so closely related to sulci. And here we should go into more detail and try to better understand what the relationship is. So let's start with the primary areas, primary motor and somatosensory area. This is a coronal cross-section. Here is the central sulcus on a very ventral level, and indeed we can see that at the central sulcus there is a border between primary motor area 6 and somatosensory areas here 3A and 3B, and also is visible very nicely on these cytoarchitectonic sections that indeed the somatosensory cortex has a much smaller thickness than the primary motor cortex. So this is also a sign that you can see, of course, in magnetic resonance images, and the border is more or less in this fundus of the sulci. With the primary auditory cortex, it is very similar. This is a mushroom-like Heschel gyrus, and at least at the surface, the primary auditory cortex can be found. When we have a more detailed look to the cytoarchitecture of motor and premotor areas and their relationship to landmarks, then we have here now zoomed into the primary motor cortex, and the primary motor cortex has a very typical cytoarchitectonic pattern, and it is clearly and relatively easy to identify in all the different brains. So what does it mean, easy to identify? First of all, there are very large cells, giant pyramidal cells in the so-called layer 5. This is the inner pyramidal cell layer. These cells are so big that they can have a length of 100 micrometer or 120 micrometer, and you can see them even sometimes without a microscope because they are so large. The cytoarchitecture of this primary motor area 4 is structured in such a way that there are layers indicated here by Roman numbers. They have a different thickness, so layer 3, for instance, is very broad, while layer 5 is a little bit smaller. Layer 6 is not completely imaged here. And layer 5 pyramidal cells, they give rise to the pyramidal cell, to the pyramidal tract, which you have seen just recently in the talk. And if you imagine that sometimes the axon of these big cells may reach half a meter or maybe even longer because they go to the spinal cord, then it becomes clear that these cells should be relatively large in size. Another characteristic feature of this primary cortex is primary motor cortex. It does not have a clear visible layer 4, which is the inner granular layer and which is characteristic of most of the other cortical areas in the brain. And the reason here is that during very early postnatal ontogeny, the pyramidal cells of layer 3 and the pyramidal cells of layer 5, they intermingle and they dissolve layer 4. So during prenatal ontogeny and early postnatal ontogeny, during the first months of life, this layer is present, but then after that in the adult brain is not so much present anymore. So this is a very characteristic feature, and therefore we call it agranular cortex because it is lacking the inner granular layer. And this is very similar to the so-called premotor cortex, area 6 of Brodmann, which is located at the precentral gyrus. It also does not have a layer 4. But if you then have a look to the other layers, it's a little bit different as compared to area 4. And one of the differences is that large pyramidal cells are here located in layer 3, while the pyramidal cells in layer 5 are not so frequent and usually they are also not so large. So there's an inverse relationship of the size of layer 3 and layer 5 pyramidal cells as compared to the primary motor cortex. And these are cytoarchitectonic criteria which help to identify these two cytoarchitectonic areas from each other. They are both very broad. They both have a thickness of about 4, sometimes even 5 millimeters. So these are the most thick or the most broad cortical areas in the brain. The border between those two areas is not identified by any sulci. So the precentral sulcus is not a landmark indicating you where the border between premotor and motor cortex is. While the primary motor cortex in the central sulcus has a clear border to the somatosensory cortex, and here you can be quite sure that you have identified the right region. When we go to the primary auditory cortex, the mushroom sign, the Heschel gyrus, is very clearly visible. And at the surface, at the free surface of this Heschel gyrus, the primary auditory cortex can always be found. This is the lateral surface, where at the superior temporal gyrus area TE3 or the Wernicke, belonging to the Wernicke region, can be seen. However, the border between the primary auditory cortex and higher auditory cortical areas is not precisely located, so to say, in the fundus of the sulci. It is more located here at the upper position, and then the primary auditory cortex borders to higher auditory area, multimodal areas. Let's have a look to the next example, which is a primary visual cortex, which is related very closely to the calcarine sulcus, but again in a quite specific way, because when you see the calcarine sulcus in an image, you can be sure that in the surroundings the primary or visual cortex can be found. However, the more lateral borders, the more dorsal ventral borders, they are not restricted or limited to the localization of a certain sulcus, and this is an example of the visual cortex coming from the occipital pole, from here You see here very nicely the transition between area V1, primary visual cortex, and V2, higher visual areas, but they are not located in the sulcus, but they are located at the free surface. And this is another example of the primary visual cortex where the border is at the free surface, and a very abrupt change in the pattern of the cytoarchitecture from V1 to V2, higher visual area and extra striate area can be found. And very characteristic for this particular area is a very broad layer 4, a very broad inner granular layer, and this is indicative of the massive input that we have in the primary visual cortex from the lateral geniculate body, for instance, which projects two neurons in 4B here in particular. And this is a little bit different in V2, which is processing further visual information. It has also a granular layer 4, but it is not as broad and prominent as it is in the primary visual cortex. So V2 is a nice example, a typical example of an isocortical, neocortical area. All the 6 layers are present, can be identified: a molecular cell layer, an outer granular cell layer, an outer pyramidal cell layer, an inner granular cell layer, inner pyramidal cell layer, and a multiform layer. And I would like to put your attention also to the fact that the border between the cerebral cortex and the white matter has a more or less sharp transition to the white matter. And this is very different in the different brain region. And the sharpness or the abruptness of the transition between the cerebral cortex and the white matter determines to a large degree how well you can see and identify these borders in magnetic resonance images. And sometimes it is very easy, like here in area V1. Sometimes it is more difficult and the cerebral cortex is more fading out. And this also creates different difficulties in the precise estimation of the identification of the cerebral cortex. And this has consequences when you segment the cortex based on MR images and at such criteria. But now let's have a look to another area which comes from higher associative cortex and more precisely Broca's region. And we heard already this is where the M sign is. There are 2 major gyri, the opercular part and the triangular part of the inferior frontal gyrus. And what you see here are cross-sections through the brain showing mainly here the opercular part. This is here. And here, this is a transition to the triangular part. There is a length mark, which is quite prominent. This is the inferior frontal sulcus. This is the most ventral of the frontal sulci. And what we have delineated here is the extent of areas 44 and 45, which are probably belonging to the core region of Broca's region. And what you see here is that the extent of these 2 areas do not match the sulcal pattern. So, for instance, in this image, the area 44 more or less stops at the fundus of the inferior frontal sulcus, while in this section from the same brain, only a little bit more anterior, the area 44 goes to the upper bank of the inferior frontal sulcus. So we are already on the middle frontal gyrus. And in this particular case, even more anterior, the area 44 does not reach even the fundus of a sulcus. And if we consider that such sulci may have a depth of 1 centimeter approximately, then we have quite some variation with respect to the fundus of 1 centimeter or 1.5 centimeter. And as illustrated here on the left side, we can also have the case that we have in one and the same coronal section here, different cortical areas are present very closely to each other. So for areas 44 and 45, the borders of the area do not match sulcal landmarks to a large degree, although we can be sure that more or less in 100%. At the free surface of the opercular part, there's 44. On the free surface of the triangular part, there's 45. But the border is varying, and this depends, for instance, on the level, on the number of the coronal section which is identified, and in addition, it also varies between between brains, but also between the different hemispheres. And this gives you an overview of the variation that we see in a sample of 10 human brains. Areas 44 and 45 are here drawn to the surface of the brain. This is a mapping of the left and of the right hemisphere, of the first brain, second, third, fourth, and so on and so forth. And you can see there's a considerable intersubject variability of the size and of the extent of these two areas, 44 and 45. And there is an intersubject variability in the size and extent of the cytoarchitectonic area. There is an intersubject variability in the size and extent and presence of sulci and gyri, and the relationship between both, that is the sulci, and the areas also vary in each different individual. But if this is the case, that there is so much variation in sulcal pattern and in the relationship to brain areas, then the approach that was selected by Tallarach and Tourneau to project Brodmann's map to a very concrete individual post-mortem brain and then provides basis for the so-called Tallarach atlas is, so to say, a very crude approach. So these authors really just transferred Brodmann's map to another brain based on sulci and gyri, but it is now clear that due to the intersubject variability, this can only be a very rough estimate. And there is another problem or another constraint that we have to take in mind. This is Brodmann very rarely described the extent of his areas with respect to sulci in detail, and he provided only a very few images. And the result is that in Brodmann's, that in Tallarach's atlas and many tools which are like this, we have quite some variation where the border is. And this is, for instance, an example of the border between premotor area 6 and area 8, which might be here or there or here or there. And again, the variation is about 1 millimeter. And we will come in the next talk to very high resolution MR imaging shown by Rainer Goebel, and you see that we are, of course, much more precise at this moment, and a precision of 1 millimeter or 1 centimeter, of course, counts to a large extent. So what is the consequence? We need a good atlas. We need a good map. This map should be precise, and this is an example of a scientific challenge with a precise map. So this is Vasco da Gama's journey from Europe to India. He had a very precise map. He was very well aware of the perimeter of the Earth, and he know how big the continents are. So he found his way to Asia very easily. And at the same time, Columbus had the worst map. He went into the wrong direction, and he ended up in America because he underestimated the size of the Europa and of the Asian continent, and therefore decided to go to the west as compared to go to the east. And you may say, well, he found America, which is quite an endeavor, but this was not precisely what he was aiming to do, and your funding organizations are perhaps not so excited if you find completely different things than you have promised in your grant application. So to have a good map is important when we want to understand the Earth. It's also important to understand the human brain. And there's another aspect of organization of the brain that I would like to emphasize, and it can be also seen already in Humboldt's map. This is, there are different aspects of organization of a structure that have to be kept in mind. And we would call it today a multimodal atlas. It is very similar here in Humboldt's map, which also reflected different aspects of organization of the of the Earth, and we can do the same and can learn for it in the brain. And one important aspect, in addition to cytoarchitectonic, this is the so-called receptor architecture, which shows the distribution of receptor types of different neurotransmitter systems, and Carl Zillis will talk about it a couple of minutes later. Why it is important for mapping? Because we can see that for different neurotransmitter systems like acetylcholine or glutamate or GABA, we see differences in the concentration of these receptors in dependence on the different brain region. And again, if you have a look to the opercular part of the inferior frontal gyrus, then we can see that the concentration at the free surface is relatively, it's a medium concentration, while when we come to the middle frontal gyrus, the concentration is going up, indicating here by the red colors. And receptors also help us to identify within the opercular part of the inferior frontal gyrus two sub-areas of area 4, a more dorsal part and a more ventral part, which differ, for instance, in the expression of this particular acetylcholinergic receptor. There's another aspect that is important when we think about cytoarchitectonic segregation, and this is we cannot base our knowledge only on visual inspection. And this is true not only for cytoarchitecture, it's true for any mapping approach. We need to identify quantitative criteria that help us to reproducibly identify cortical areas. And we did this here, for instance, in the frontal pole. This is Brodmann's area 10, where we can see when we analyze it under the microscope that there's a border between two different areas, the frontal polar area 2 and the frontal polar area 1, which are different, for instance, here in the density of the third layer, the outer pyramidal cell layer. And this difference in the cytoarchitecture can be measured using statistical methods and image analysis, and we can identify peaks where the cytoarchitecture changes significantly, and these peaks match our visual inspection and our observation with respect to the microscope. But the big advantage is that we can really make it reproducible and find the border always at the same place. And this is a method that we are using in order to map different brain sections, 3D reconstruct them, and these are 3D reconstruction of 3 out of 10 brains, which can then be brought into a common reference space, which is important in order to compare then with images coming from MRI, for instance, or from PET. And we can superimpose the individual maps in such a way that they can be represented as so-called probabilistic maps. And the probabilistic maps show for each voxel of the reference space the probability with which a certain cytoarchitectonic area can be found. These are the two areas, the middle, the more mesially located areas, FP2, and the more laterally located area, FP1. And these two areas have an intersubject variability. Also, other areas, for instance, Broca region, have a higher variability, and third types of brain regions, for instance, subcortical nucleus, they have very often a lower intersubject variability as indicated here. Based on these probability maps, so-called maximum probability maps can be calculated, and these are examples of maximum probability maps of different brain regions. So this is the primary visual cortex. Here we have the premotor cortex, the frontopolar area 1, and these areas are available for download. They can be used to better localize functional imaging data. Unfortunately, it's very time-consuming to calculate such maps, so at the average to identify one area in 10 brains, one needs approximately 1 year or 2 years if the brain regions are very difficult and if the areas are large. But then these areas can be used by other groups in order to superimpose different aspects of brain organization. And the HPP atlas is an atlas which allows more and more to online analyze different cortical areas with respect to different aspects of brain organization. And one of the challenges that we had to overcome in order to develop such an atlas was to make it online and to make it easy to use. Here you can see some applications of this atlas. For instance, in the common reference space of the MNI format, you can see different cytoarchitectonically identified areas labeled here by different colors. And it is possible to cut the brains at different levels, also to introduce oblique sections, which is very nice and a very nice tool, because sometimes a folding pattern do not allow you to see the areas in the best way when you have only coronal and sagittal section. You can play around with them. You can have, for instance, cytoarchitectonic maps, probability maps, here indicated with a seed scale, or you can also see maximum probability maps. And then for each of these areas, different types of information is available according to the brain region, according to the areas. There's a very intuitive way, for instance, to go into the receptor architectonic organization, and you can extract information, for instance, about the distribution of the receptors from the cortical surface to the white matter borders, quantitative information which characterizes a certain brain region, receptor fingerprints, Calcilis will show it a little bit later, can be identified. And the task is now that we populate the brain atlas step by step by such type of data. Also, for instance, by data coming from electrodes, from patients with epilepsy, where we would like to include dynamic information extracted from these electrodes. And the idea is really to have a multi-level, multimodal atlas and to bring together the different types of information coming from different labs. And you will not find all the information, but as I said, more and more will be included. One of the tools that is there is a so-called UJEX tool, which helps to analyze the relationship between gene expression and probabilistic cytoarchitectonic maps. So the gene expression data comes from the Allen brain atlas, the cytoarchitectonic maps comes from the Jülich atlas, and when both types of maps are being superimposed, you can ask question which specific gene is expressed in a certain cytoarchitectonic area. And this allows you to make conclusions, for instance, about differences in the expression of certain genes that are important for receptors, for instance, or that are changed in patients with major depression, for instance. So this is a type of tools that we are providing more and more in the context of the Human Brain projects. So is this enough to have this type of atlas? Of course, we would like to go to a level of detailedness that allows to image every single cell as we can see it here. But then we have to capture a very large amount of cells. So each human individual brain has about 86 billion nerve cells. And if you want to go to 1 micrometer resolution, we have to handle very large images and come up for one brain with 1 petabyte space, which is quite a lot and which poses not only neuroscientific challenges, but also challenges in terms of data science and data analytics. But it is feasible to handle such large sections, and the feasibility is illustrated here, where we have a section through the brain, and these are virtual sections where we have 3D reconstructed on a cellular level the distribution of cell bodies in 3D. And you can see that indeed it is possible to 3D reconstruct single cells and to find single cells which are physically cut when we are processing them in a histological way. The problem or the challenges are similar when it comes to fiber architecture, and Svenja Kaspers has illustrated some examples We are using in our lab 3D polarized light imaging in order to analyze sections, and very often when we use such very high resolution methods like polarized light imaging, like two-photon imaging or light sheet imaging, we can only image small pieces of the brain. This is different obviously in rat and in mouse brain, but human brains are really big. And then it is important to have a tool which allows to integrate data coming from ultra-high resolution analysis into the big brain context and to navigate. And this is done by a very nice tool, which is now also available. And this allows, for instance, to register data coming from ultra-high resolution imaging, like 3D polarized imaging, in the context of the so-called big brain data set, which is one of the templates that is supported by the Human Brain Atlas. And you can see here the big brain template with different, with a high level of spatial resolution, 20 micrometer spatial resolution, and we can go to different brain regions, and all three orthogonal sections can go also further to the subthalamic nucleus, as can be seen here, and then register data coming again from ultra-high level spatial resolution of the subthalamic nucleus to this particular spatial template. And this is a little bit the idea how we conceptualize future human brain atlases to combine the different types of information on the different spatial scales with respect to the different modalities in one and the same reference space, in order then to come up with better concepts of what is the relationship between sulcal landmarks and microstructural areas. So if you let me to summarize, cortical areas are functionally relevant. You have seen some of the examples. We see that the properties of neurons change when you come to a neighboring cortical areas, but the borders cannot be precisely predicted from sulci and gyri. There are different relationships. For some area, there is a straightforward relationships. For other area, it is much more complex, and the sulci are not good indicators. And this, of course, is a challenge because we usually want to understand the whole human brain and not only certain areas. The classical Brodmann areas underestimate the number of cortical areas, and the two areas in the frontal pole, FP1 and 2, they are two examples of many. We expect that there are approximately 200 or even more cortical areas in the human brain as compared to 40 in Brodmann's map. Then we need multimodal and multi-scale approaches because one modality never explains the whole story, and we want to have, of course, always understand what is the relationship between the different functions of a brain region and its structure. The cortex is highly variable. Yeah? The big brain model helps to include cellular data into a new reference space and to compare different types of data. Fiber mapping at microscopical and mesoscale scale is possible using PLI, and it helps also to better understand diffusion data. And the Human Brain Atlas is a new type of atlas that helps to integrate all these types of different data. Yeah, thank you for your attention.